Hi, welcome to Detours, Understanding Acquired Brain Injury. And this is the third section in Neurogenetics Introductory, where we're looking at things that go wrong with chromosomes, with genes, epigenetics, and we're going to talk about a few of the reasons that chromosomes can fail. And then we'll be getting into genetics shortly. An error in cell division is definitely the most common cause of chromosomal disorders. And the reason for cell division failures can both be environmental or an inherited disorder. Inherited disorders are not common, as people with chromosomal abnormalities rarely reproduce due to the number of cognitive or behavioral disabilities they have. It happens, but it is possible to be a carrier. We talk about this in genes and chromosomes, where a disorder is carried, and we will mention it again, but somebody with a full-blown disorder like this that was likely to be inherited usually has too many disabilities to have children. I mean, there are tragic circumstances, of course, but typically that's not. More often it is due to a de novo problem where it spontaneously arises either before or after fertilization of an egg that these kind of chromosomal disorders happen. There are milder versions of chromosomal abnormalities, and people with the milder versions can reproduce. They live fulfilling lives, and, they go on to have kids and all that kind of thing. But serious chromosomal disorders usually are life-threatening or severely permanently disabling. I mean, that should be obvious, but, So what are the causes from the environment? Well, typically, age of the parents. Usually this is more often associated with mom, but with a male, with the father, typically what happens is the number of sperm drop off, and so they're just less likely to be able to fertilize. Can they pass along an error? Yeah, absolutely, no matter what the age. And we're seeing a growing number of younger men and women have kids that have chromosomal disorders. And that relates to the next items, which is exposure to toxins, radiation, an environment with things in it that just, are more likely to cause this. Women are also more vulnerable because they produce the majority of their eggs in the initial divisions while they're developing in utero and while they're still young. The eggs do not finish the cell division process until the woman's ready to ovulate. So if the egg is produced, though, that is aneuploid in an early stage of cell division, it's likely to not be ovulated till later because it is heavier physically. So it tends to sink. So it will be, that is why an older age plays a role, because that second set of chromosomes, that second chromosome will make it. However, it still can be. Usually a younger woman will delete the aneuploid eggs as well, whereas if she's older, the body may not find and destroy these eggs. Remember, we discussed checkpoints in chromosomes and cell division, chromosomes and genetics. And what's interesting lately, with the larger number of women having aneuploid pregnancies, it suggests that these other factors play a role. So there are, other things to, keep in mind here. Toxins from nature, artificial sources play a role. So radiation exposure we're talking about typically ionizing radiation and all different kinds, whether it's medical diagnostic imaging, radiation therapy, whether it's, flying, internationally, high altitude very frequently. Perhaps your job involves ionizing radiation. Do you know? Check out our video on the atomic genie in medicine and acquired brain injury. Nanoplastics and nanoscale metal found in cosmetics, water, even in the air you breathe, I stress may, cause chromosomal damage. We still don't know for sure, but, it's like a study from 2 years ago found microplastics in every placenta that obstetricians were examining. I can't believe that there's no effect from this. Heavy metals, volatile organic compounds. If you're planning to have a baby and you or your partner exposed to environmental risk or may have a chromosomal disorder, you should consult with your physician. Maybe consider genetic counseling. These things can cause specific genetic errors too, not just chromosomal and structural diseases and disorders. So it's always smart, based on, your job and the environmental risk factors and things, to at least seek out and maybe have your own testing. What you do from there, obviously, children with these problems, these genetic errors, these chromosomal errors can still go on to live happy, fulfilling lives. So, this is one of those difficult ethical decisions. I've used the word complete when it comes to structural chromosomal disorders because there are various types of incomplete structural chromosomal disorders. Let's talk about them. Mosaicism. Mosaicism occurs after an embryo is formed as euploid. So, sperm and egg, healthy. It's got 22 pairs of autosome and is either XX or XY. Despite that, as the new embryo is undergoing its own cell division, something goes wrong with that egg, or with that zygote, that newly formed. So it's got nothing to do with parents or heritability or any of that. Something goes wrong inside that newly formed individual. This is likely what happens when this is similar to what happens when a body cell has the problem. Thing is, this is so early that it is as serious as any other aneuploidy from sperm or egg. An embryo during very, very early mitosis may have some cells with an extra chromosome because of a problem during its own cell division process. Usually it hits the checkpoint and will destroy that cell and others. Sometimes it doesn't. And so it will lead to some kind of de novo error spontaneously arising. So usually what will happen is a trisomy, an extra chromosome of one. If it is one of the non-survivables and it gets past the checkpoint, there'll be an early miscarriage. Mom may not even know she was pregnant. But in some very incredibly rare cases during very, very early embryonic development, there are other things, such as one embryo may absorb another developing embryo that has a chromosomal aneuploidy. This is not a formal diagnosis, but has been referred to as chimeric Down syndrome, specifically if it's a 21st. But there are chimeric Pateau or chimeric Edwards. Really not much is understood about this, but it's one of those kind of not well understood. But going back to where early developmental cells have a malfunction and develop this, we refer to it as mosaic, because not all the cells had this error in this early developmental stage. So only some will exhibit this. We call this mosaicism because, for example, if you look at the brain, some neurons will have the problem associated with that extra chromosome. Some won't because some are euploid. and others are aneuploid. And that means that they may not have the intellectual disability. Perhaps only, 1 out of every 25 or 1 out of every 1,000. It depends on where in the cell division stage this happens. And this is why you will get some kids who look like they have Down syndrome and may have some of the problems, but may be average, above average in intelligence, or only mildly below. Some may only look there are other cases where they don't look like they don't have the typical epicanthal folds of the eyes, and they don't have, a lot of the traditional features. But they have there's a well-known case. A woman had multiple children with Down syndrome. They checked her, and they found out she had it too. And that's why, all of her kids had it, because she also carried a trait, but she's a mosaic. And she's probably, very high order. So very, very, very few of her cells. But likely the cells that make up her ovaries and that make the eggs, all of them have it. Meaning that every child she has will exhibit Down syndrome, and full Down syndrome at that. Such is the nature of mosaic. Now, partial aneuploidy, the situation where just a little piece of a broken chromosome in a cell is either added or is missing. These tend to be, again, less destructive, but can lead to non-viability depending on which segments. Again, you can get partial Downs, partial Patau, partial Edwards. And for deletion, you can get partial Edward or partial Turner syndrome. You also can get, as I said, like for the 16th chromosome, you can get trisomy, partial trisomy 16 and partial deletion of the 16th chromosome. And there are others, again, not well known. Let's talk about something else. Sometimes a very, very, very small segment can be deleted or duplicated. We're talking about really small. So we're almost to the point where it's genetic, but not quite, because you're usually missing more than just 1 or 2 genes. You're missing a couple, maybe a dozen. And we refer to these as microdeletions or microduplications. Again, these are either inherited or occurs de novo mutations, spontaneous. So a microduplication typically happens when a few genes are duplicated in a chromosome. It's not quite the same situation, obviously, where an entire chromosome is duplicated, like aneuploidy or refuses to migrate, but it depends on which gene or group of genes are duplicated. Microduplications really are not obvious easily, and they're often missed as a case for many diseases. And these diseases weren't even thought to be necessarily caused by genetics or what have you. These are more recently discovered as we are unlocking the secrets of neurogenetics, genetic disorders, that kind of thing. Microduplications often cause intellectual disability. Autism is thought to be one of these things that's due to segmental chromosomal errors, microdeletions, microduplications. Learning disabilities, emotional, mental health problems, microduplications, microdeletions. So when we get deeper into specific conditions or syndromes cluster of symptoms. And as a result, it's like they can be classified in many different ways, but again, segmental chromosomal errors. So a microdeletion is definitely survivable, just like a microduplication, much more survivable, and often with much milder symptoms, and leads to things where you're just like, I don't know what we have, but it's like often likely to be even missed by physicians or whatever, or treated as, just They have something. As we learn more, and it may not even have a label, just idiopathic intellectual disability, idiopathic learning disabilities, idiopathic autism, whatever. And it may be due to a specific microdeletion or microduplication. And it doesn't have a name because enough patients have not been found to sample it. It may be unique to you as an individual. Quickly, just back to fragile sites are caused by a failure of chromosomal repair systems, environmental toxins, radiation mutations. They can cause, again, learning difficulties, visual impairment, motor disabilities. They're damage and structural errors, the reflection of a lot of different kind of stressors, cellular dysfunction. They kind of look like constrictions and they look like this. And the problem is that they're likely to break off and go out on their own. And with it, that leads to partial deletions or microdeletions. Next, we will be talking about specific genetic errors. This is on a much smaller scale and is problems at the gene level. We move from problems with the cookbook to problems with the recipes. Remember, genes are sequences that spell out specific traits or things along those lines. They generally follow Mendelian law, but they also can be de novo, just a random change, whether it's in a single codon, whether it is a single nucleotide, or whether it's a problem with the entire gene sequence. These are problems with the recipes and not with the cookbook. See you next time. Please click like, subscribe. We'll see you hopefully shortly, and please click like, subscribe. Don't forget to hit the bell icon and share this with anyone who might be interested in these kind of topics. Neurogenetics will, after we're done discussing how things can go wrong generally, then get into specific diseases, disorders, syndromes, that kind of thing. Bye.